The economics of feeding Reassure precision-release choline. Reassure is fed during the transition period, and because it's fed for such a short period of time, it costs just $15 per cow. And yet the benefits will continue to generate income throughout the year. Cows fed Reassure produce 5 pounds more colostrum, which pays for your Reassure investment on the very first day of lactation. Cows fed Reassure also produce 5 pounds more milk per day, every day. That means after the first day, every day is payday. Invest in Reassure during the transition period and recoup your investment on the very first day of lactation. After that, you got it, payday. Today, we welcome Dr. Daryl Nydam from Cornell University and Dr. Stephen LeBlanc from University of Guelph to provide insights into the strengths and weaknesses of research studies to help better understand the health and management of dairy cattle. Dr. Nydam is the faculty director for the Department of Public and Ecosystem Health at Cornell University. He earned his BS in biochemistry at the University of New York, Geneseo, a DVM from the College of Veterinary Medicine at Cornell, and a PhD in epidemiology at Cornell. While living in Vermont, he entered private clinical practice. He is now employed at Cornell as professor of dairy health and production and co-director of the Summer Dairy Institute. Daryl's a member of the Quality Milk Production Services, the section of ambulatory and production medicine, and is faculty director for the Cornell Atkinson Center for Sustainability. He's published more than 150 peer-reviewed papers with gifted students. Daryl is a member of the graduate fields of animal science, epidemiology, and biological and biomedical sciences, where he advises PhD students. Dr. LeBlanc is a professor in the Department of Population Medicine at the University of Guelph and director of Dairy at Guelph, the Center for Dairy Research and Innovation. He received his BS in animal science from McGill University and a DVM and DVS from the University of Guelph. His research focuses on dairy cow metabolic and reproductive health and management, precision technologies, and antimicrobial stewardship. With graduate students and collaborators, his work has resulted in 200 peer-reviewed papers and invited talks in 20 countries. He is a senior editor for the journal Dairy Science and serves as a member of the board of directors of the American Dairy Science Association. And now we'll first start with Dr. Nydam. So Dr. Nydam, welcome to the Real Science Lecture Series, and the floor is now yours.
Well, thanks a lot for having me, Scott. And it's obvious we're really lucky to have Dr. LeBlanc with us here today after that introduction. Holy smokes. So to all of you out there in Zoom land, we'll try to zoom through the next 45 minutes of your life and then get to some questions at the end. What Stephen and I hope to do is give you a high level, probably about a 2-semester course in study design in the next 45 minutes. So Epi 101, and we're going to tune in a little bit to causes and consequence and how we can understand this using different types of studies in dairy cow health and performance.
So part of what will give us a framework for what we're going to talk about today is trying to get some clarity around questions that we're asking, depending on the hat that we're wearing in our different roles in the dairy industry, and how different study types can help to answer different kinds of questions. So we'll go, very, very highbrow to begin with, and Socrates' famous questions from philosophy and science were: is it good? Is it true? Is it good? And is it useful? And we're going to borrow several of those as we go through the presentation today. Classically, a scientist's questions might be, what's the mechanism for how things work, and what's the physiological relevance of associations and mechanisms that are identified? And finally, what the umbrella that we all work under in different ways are the dairy advisors and managers questions, which might be, is doing something, whether that's a feed additive or a management practice, profitable or helpful to my operation? And all those former questions can certainly help to inform that. But at the end of the day, those questions are a little bit different, and different studies can help us get different pieces of the picture, and therefore help answer different questions for different uses.
So when we're looking at new information that gets out there, we have to understand the kind of study from which it came. And there are two big buckets out there: descriptive studies and analytical studies. And in these descriptive studies, there's really no comparison group. And there are things like case reports, case series, or surveys. So a case report might be the first case of H5N1 in a cow. Case series might be describing what happens to those group of cows, the first 5 of them. And then we might do a survey and say, well, how much of this is out here and what does it look like? And those are important pieces of the scientific process because, for things that are new or newly hypothesized, we just describe those things. But it doesn't really help us get down to where there's a strong association with some management factor that we might wish to employ, or even a causal mechanism. So we're not going to talk about these kinds of studies anymore today, but they are still good. What we will spend time on in dissecting and digging into a deep example of one sort of transition cow issue is analytical studies. And analytical studies, their hallmark is that they have a comparison group, and that makes them different than those simple descriptive studies. These analytical studies come in 2 major flavors: those which are observational and those which are experimental. The observational ones, you don't fiddle around with the study subjects. You just let nature take its course and then try to rigorously describe it. And that has upsides and downsides. In experimental studies, you fiddle. Those are interventional studies. Among these observational studies, sometimes mislabeled collectively as epidemiologic studies, and sometimes lumped all together, but there are 3 important ones, and we're going to go through them a little bit deeper with diagrams. But there are cross-sectional, cohort, mostly prospective, and case control. And the major difference there is when in time the exposure or treatment is measured in relation to the outcome or perhaps disease. The big difference between, as I mentioned, observational and experimental is how the study subjects are allocated into groups. Often in randomized trials, we'll randomize those study subjects into groups.
So for today, we're going to use ketosis or hyperketonemia as an example that we'll lean on throughout most of the talk to delve into and dissect some of the questions and the study designs and answers that we can get from those studies in the context of ketosis. So just to be clear about what we're talking about and try to use precise words for things, hyperketonemia is essentially what the word implies: high level of ketones in the blood. So as far as that goes, it's just a description, no more or no less. To a point, cows mobilizing body fat and oxidizing it to produce ketones as an alternative fuel to allow to spare glucose to make milk is very much a physiologic thing. Cows need to do that to make lots of milk. So physiologically, that could just be describing ketonemia. But at some point, through many observational studies, it's been established that at some point, the level of BHB pumping through a cow's veins may get to a point where that's at least associated with undesirable consequences later on. And so that could then be classified as hyperketonemia because it's associated with greater disease risk or culling risk or less milk subsequently. Not necessarily to say consequently. Clinical ketosis, again, just as the name implies, clinical in a medical sense means visible. So it means an animal that has hyperketonemia and also has visible clinical signs, like being off feed, for example. It's worth pointing out that there isn't really any consistent threshold of blood BHB that's associated with the appearance of clinical signs. And last but not least, subclinical, again, means you can't see it. There aren't visible signs visible to naked eye. So if you stood next to a cow with hyperketonemia or subclinical ketosis, she wouldn't look abnormal. But if you measured her blood, it would be in this risk category, whereby her risk of some subsequent badness or undesirable condition is greater than it would otherwise be if her blood ketone levels were less than X. And we'll come back to this a few times through the presentation, but this is maybe a classic example of, in the context of dairy cows, of the fable or the parable of the blind men around the elephant, whereby each observer is correct, or at least not wrong, in what they perceive when they're considering their part of the picture, but none of them necessarily has the whole picture. And so, that leads to the classic answer, does it matter, or what does it do, or how does it work? It depends. And absolutely it depends, but we can use a variety of study designs to help us actually answer it depends on what and in what circumstances. And lest there be any confusion or worry that observational studies or epidemiology in general is a bit of a data free-for-all, there is quite a bit of rigor and structure to trying to sort out what is associated with what, or even what causes what. And there are ongoing, very big, very rigorous debates in epidemiology about whether observational studies can truly establish cause or not. I'm not sure there's a definitive answer to that, but one of the classic ways to bring rigor to this is employing Hill's criteria. So those are named after Sir Bradford Hill, who was a very famous medical epidemiologist who published this classic paper back in the 60s. And out of that came Hill's criteria for causation, or at least association. And we won't go through all of them, but a number of them we will touch on today. And those include things like, in observational studies, the strength of the association with the outcome of interest, the consistency of that association across studies, the biological plausibility of that based on what we know, which links to experimental evidence that would also support it. And very importantly, temporality. In other words, time ordering. Does the exposure, does the risk factor come before the outcome of interest in time? And all of these and a few others can be combined to build the case, build the strength of evidence that something contributes to, perhaps even causes, but at least is rigorously associated with an outcome of interest. And so going back into the 1980s, 1990s, there were many studies published that included path diagrams, also called causal diagrams, which might be part of a source of some confusion about what we're talking about. But these illustrate associations among, for example, in this case, transition cow health disorders. And it's worth remembering that back in the 80s, for example, this was novel, this was really useful and helped advance the understanding that these things were connected. They didn't just occur in isolation, and in generating hypotheses about how they were connected, and contributing to an iterative process of finding associations, looking for mechanisms and so forth. And that wheel keeps on turning. So again, in this one example that's on the screen, they've done a pretty good job of getting things about right in terms of time ordering and temporality. These are associations that have been observed across a number of different studies, but it's not to say that they're causal in a very direct, mechanistic, physiologic sense. And so one of our themes throughout the rest of this presentation will be that what is useful to the advisor, to the manager of a dairy, is not necessarily causal. Sorting out what's causal is generally informative and interesting. But again, we don't necessarily need to establish causality to have utility. And things are complicated. And so if we only, no one study can do it all. That's just not practical, not feasible. But it is worth remembering that whether we're looking for associations or even causes, these things are a little bit complex. So, does disease cause death? Yes, it can. Does disease cause culling? Yes, it can. But is it the fact that the disease in a very literal or mechanistic sense caused the cow to be removed from the herd? Or is it that disease caused the cow to eat less, therefore milk less, and therefore eventually be removed because her production is low, and so on and so on. And this is, I think pretty intuitive to people working in the dairy industry, whether that's in the field or in research. But it is worth pointing out that it's tough to do one study that adequately captures all of these dependent, interacting, intervening variables, because it's complicated to collect all the data, collect enough of it, and make sense of what can be some pretty complex pathways linking these things. And last but not least in this segment is survival bias. So there are many kinds of bias that can creep into studies despite our best intentions. But one that's worth mentioning here is survival bias. So essentially we only measure certain outcomes on the survivors. So if we look at how much milk cows made across a whole lactation, the cow has to make it through the whole lactation to be included in that number. That's even true for peak milk or week 4 milk. There will always be some survivor bias in there. And so we should at least keep score of how many cows started the study, went through the transition period, but were culled or removed or excluded before our outcome of interest, whether that's milk or pregnancy, for example. And with that, I'll turn things back over to Daryl to go through a number of the types of studies that can help us answer these questions that we're interested in.
Cool. Thanks, Stephen. Now back to the boring nuts and bolts after some nice philosophizing. So I clipped these really nice pictures from our colleague Dirk Pfeiffer. And you can see here one of the kinds of analytical studies, often misclassified as an epidemiologic study, but this is the classical cross-sectional study. And it answers the question, what's happening right now, i.e. what might be the prevalence? So there we take a sample, in this case, a bunch of cows, and we're going to measure at the onset of the study, both the exposure or the treatment and the outcome at the same time. So we can establish some associations, but we don't know truly in this kind of study which came first. Chickens or eggs came first, matches, smoking, or lung cancer came first, hyperketonemia, displaced abomasum. We don't know which came first. Perhaps we don't even know some immune outcome. They all happen at the same time. However, if you just need to go out to a farm once and get this, these can add some good value to our scientific knowledge. Here's another kind of analytical study, which we use when we have rare events. And in this instance, we select study subjects based on whether they had the case, and then we get some controls to go with them. And then we look backwards in time to see if they had the exposure of interest. These are efficient studies, as I mentioned, for rare outcomes, and we're not going to belabor it any further. So here's one we want to dig into a little bit more, the prospective cohort study. So again, this is an analytical study that is not interventional, right? So we have a cohort, in this case, cows, and at the onset of the study, they are either exposed or unexposed. and it could be exposed to semen or not semen. They could be exposed to being hyperketonemic or not hyperketonemic. They could be exposed to having metritis or not having metritis. And then you'll see the onset of the study and the timeline going to the right, as well as the direction of inquiry, answering the question, what will happen? In studies that are conducted well in this manner, the prospective cohort study, all of these exposures are time-ordered and precede the outcome. The outcome could be premature removal from the herd, displaced abomasum, high or low milk production, getting pregnant or not. And this is the prospective cohort study. It gets us closer and higher up in Bradford-Hill's criteria because we do establish this temporality. And now, what I think is near the top of the pyramid of analytical studies are the randomized controlled trials. And you'll see the geometric shapes here look just the same as the prospective cohort study. There's circles and there's rectangles and diamonds. So here at the start of the trial and time going forward, we have a sample or a cohort, which are then divided up into treatments and controls, or exposures and non-exposures, that we follow forward in time to see if they have the outcome or the disease. Could be metritis, not metritis, high milk production, low milk production. But the difference between this and the prospective cohort study is that the subjects were not naturally put into two groups, exposed and not exposed, but we randomized them. And the great thing about randomization is it allows us to allocate, if the gods of randomization are with us, allocate a whole bunch of unmeasured potential bias equally amongst the treatments and the controls. So those things distribute nicely. It actually helps sometimes with doing fancy statistics. If this works out well, you don't need to do a lot of fancy statistics, though you probably still should. In prospective cohort studies, you have to use fancy statistics to control for all sorts of post hoc bias. So here's just a summary of these three kinds of analytic observational studies, moving from cross-sectional to case control to prospective cohort. All of them have bias, as do randomized controlled trials. And you can see here how they're divided in sampling. We get a random sample of the study population. In the case control, as I mentioned, we get cases and controls and then go backward in time. And then the prospective cohort study, we have exposed and not exposed that are followed up for a specific time going forward, as opposed to the cross-sectional study where it's all done at one point in time. And then taking up this concept of causality, in the cross-sectional study, with a lack of temporality, we can only establish associations. They could be very strong associations, relative risks or odds ratios of 5, 6, 10, right? Things you'd never hear of in human cancer studies. However, in the prospective cohort study, because we're quite certain that the exposure preceded the outcome, we're getting, again, higher up those Bradford-Hill's criteria of causality. And then there's a number of measures we can look at to compare the risks, and these are, at least in cross-sectional studies and cohort studies, relative risk, risk ratio, odds ratio. With a little bit of simple math, you can convert between them, so it shouldn't give you any trouble that some studies use odds ratios and others use relative risk. So it's pretty important to know, when we're trying to make decisions about what's causal or what's useful, where we are on this pyramid of evidence. And I'm going to submit that it's all evidence-based medicine or evidence-based production. And when you're making decisions, you should just know where you are. Am I basing this off of first principles and bench research? Am I basing it off of Dr. LeBlanc's clinical experience? He saw 2 of these when he was a young veterinarian, so therefore he will proceed thusly. Or are we moving higher up this pyramid where we're using case control studies or cross-sectional studies? Or are we getting higher to where we're using small randomized trials or prospective cohort studies? Or even getting higher where we're using large-scale randomized commercial field trials? right, we're then going to have, and we're going to get into this ideas of internal, external validity. But as we get higher and higher up this pyramid, hopefully we're having still really good internal validity as well as good external validity. And then sometimes we can mash up a whole bunch of studies, whether they be small pen studies, cohort studies, or randomized trials, and put them into systematic reviews, and then do some appropriate analyses and call those meta-analyses to get even closer to what the universal truth might be. So let's step back, and if Stephen allows me to philosophize a little bit, this is usually part of his shtick. Ask the question, does smoking, that would be cigarettes or whatever else you want to smoke, cause cancer? Or merely can we say that smoking is associated with cancer risk? To my knowledge, there hasn't been, and I really doubt there ever will be if it hasn't been done already, a large-scale randomized controlled trial on the effect of smoking and the incidence of lung cancer. Partly because if we wanted to get people to try out for this study, you might have an argument about who wanted to be in the Winston group and who wanted to be in the Marlboro group. And we may have some selection bias there, or recall bias even. There's further unlikely to ever be an experimental challenge about the particular chemicals in cigarette smoke, right, to isolate which might be the cause of cancer. So we have to ask ourselves, do you want to start smoking in the absence of randomized trials? That's a personal decision. So let's step, something that's not quite as silly, right? It's obvious we're not going to do randomized trials on Winston, Marlboro, or not smoking at all. But how about parachute use to prevent death and major trauma related to gravitational challenge? So at the top of that pyramid, right, are systematic reviews and appropriate meta-analyses. So our good colleagues, Smith and Pell, and they published this in the leading biomedical journal, the British Medical Journal. So they went out and looked for all the randomized trials for parachutes, death, and major trauma. And if you can read the fine print there, no randomized controlled trials of parachute use have been yet undertaken. So the basis for parachute use is purely observational. And apparently the efficacy could be potentially explained by a healthy cohort effect. So I'm still not sure if I'm going to use a parachute when I jump out because it has not yet been proven to be causal to protect me. These researchers here from Harvard, UCLA, and our good colleagues at the University of Michigan looked at that meta-analysis and found there were no randomized trials. So they set out to do one. And here, with great internal validity, they did a randomized trial on parachute use to prevent death and major trauma when jumping from an aircraft. So they went, and if you read the fine print there, they found parachute use did not significantly reduce death or major injury. 0% in the parachute group and 0% in the controls, with a very impressively large p-value of greater than 0.9. Very, very unlikely due to chance, that lack of difference there. So what might explain this? If you read a little further, right, that when they screened people to get into this, they couldn't get anybody to be at the altitude and velocity of some things, the altitude being 9,000 meters on average for non-participants. However, the participants in this study jumped from aircraft at an average height of about 0.6 meters. So those of you who are from backwards countries like me, that's a few feet in the air, like the young lass here. Again, even though towards the top of the pyramid, randomized controlled trials, even with great internal validity, which this study had, they randomized the subjects to treatment group. They were controlled very well for information bias. There was really not a lot of selection bias. However, the external validity for this is probably not what we're looking for, right? It's kind of like doing, we want to investigate something about transition cows and we use steers, for example, right? Just a different population. Doesn't mean it's a bad study, it just doesn't help us get to the heart of the question. So as I mentioned, when you're reading studies, you want to ask yourself about validity. First, internal validity, and that is basically, was the study done well? Were the outcomes measured accurately? And did they work through all these different kinds of bias, like selection bias, information bias, confounding bias? And if the study is good, i.e. it has good internal validity, then you might going to ask your question, is this generalizable to my situation? Is the study population relevant and comparable to my interests? So here's an example of a recent review on the subject of transition cows, which I've plucked out a couple excellent biochemical figures, which I even use in teaching now, about the biochemistry of hyperketonemia in early lactation cows. And in this study, they do point out, in this review rather, that there are different kinds of studies, some of which are randomized, some of which are observational. And here they're particularly looking at ketosis. And if you see down there in the B panel of this, they posit that ketosis has a two-way dotted line with disease, and they note that ketosis and disease occur contemporarily. And that sometimes happens. However, many of the studies were not, that it's postulated this strong association, were not cross-sectional studies, but rather prospective cohort studies. And even in any kind of study, we have to think about the risk of making a type 1 or a type 2 error, and some of that has to do with sample size. So in measuring reproductive outcomes or any other dichotomous outcome, which repro being a pretty important one in the dairy industry, let's take the example here of a difference between 10% in one group and 20% in the other group. And I went through and happily calculated a bunch of 95% confidence intervals for you. And let's, for example, first take 2 out of 20. So we randomized 20 cows to one group and 20 cows to another group. And we had a twofold difference, 10% and 20%. You can see the imprecision in those confidence intervals. We can only with 95% confidence say that the answer was truly between 1.2 and 32%, or between about 6% and 44%. And on we go up. With a difference of 10% in dichotomous outcomes, we have to get to about 300 study subjects or 300 cows in each group before we can say with 95% confidence that those two proportions are different than each other. So that's a lot of cows, and it presents some study challenges. So just be aware of that. To belabor the point, only a 5% difference, and I relax the confidence interval to 90%, we have to get up to 600 study subjects or 600 cows in each group before, with 90% confidence, we can say this 5% difference is beyond chance. So the classic 30 cows in each group, if you're measuring things like displaced abomasums or death or reproductive outcomes, and someone tells you they didn't find any statistical difference, look at the numbers and ask yourself, was there enough power to detect a difference? Just to illustrate the difference with continuous outcomes, here's milk production, and you can see that the precision of the estimate or the confidence interval is much narrower when we have continuous outcomes like milk production or perhaps some biochemical marker. So just here it is, the width of the confidence interval affected by the confidence you want to have in it. So you could be 95% confident, 90% confident, 80% confident, and that's going to change the width of that confidence interval. And then the sample size. As the sample size gets greater, the precision of your estimate gets better and better, and that confidence interval gets smaller. And then for continuous outcomes like milk production or some biochemical marker, the standard deviation or the variability around that, if there's a lot of chaos in the data, that standard deviation gets bigger, the imprecision gets bigger also. So my last little rant here is about p-values, because all the papers written in science have p-values in them, even the ones about parachutes. So I believe the p-value is a very, very useful statistical measure. However, it's commonly misused and misinterpreted. What the p-value is simply the probability under a specified statistical model that the summary data, which could be two sample means, for example, average milk production, or two proportions, displaced abomasums, in two compared groups, would be equal to or more extreme than its observed value. Statistical significance, or low p-values, is not equivalent to scientific, human, or more importantly, or at least as importantly in dairy production medicine, economic significance. Small p-values do not necessarily imply the presence of a large or more important effect, and larger p-values don't necessarily imply the lack of an important effect. Any effect, no matter how tiny, can produce a small p-value if the sample size or precision of the measurement is high enough. And large effects may produce unimpressive, i.e. very large p-values with small sample sizes or imprecise data. So always ask yourself, what's the risk of a type 1 error? Saying there is a difference and there really wasn't one? Or a type 2 error, did they conclude that there's not a difference when there really was one? Both important errors that happen in science. The type 1 error often related to that p-value, and the type 2 error often related to sample size. So just one quick example. Here's a study that we did with excellent internal validity on feed additives. The study was done well, and when we did feed additive 1, we realized 5 pounds more fat and protein corrected milk and increased dry matter intake commensurate to support the milk production with a p-value of 0.07. And this feed additive cost 5 cents per cow per day. We didn't see any other differences in other important bioeconomical outcomes like reproduction or somatic cell count or greenhouse gases. And then we had additive 2 that was 2 pounds more fat and protein corrected milk with a p-value of 0.04. Yet it cost 20 cents per cow per day to feed. There's some more math to be done before we decide which one of these is going to be better to feed. In fact, I might accept this type 1 error risk of 7% for only expending 5 cents per cow per day to realize 5 pounds of milk. I might not want to take a lesser type 1 risk of 4% to feed something that costs 20 cents per cow per day and only get 2 pounds of milk. Just more math to be done to think through.
So as we make the clubhouse turn in our webinar this morning, want to turn our attention to a couple of really good review papers that have been that have constructively set off a lot of good discussion and debate. The first one was published a few years ago in Journal of Dairy Science by Aaron Horst, Sarah Kavidra, and Lance Baumgard. And they went through quite an extensive discussion of transition cow biology and issued some challenges around making sense of some of the kinds of data and associations that we've been talking about today. And so we're in strenuous agreement that correlation is not causation, that association is not causation in and of itself. But there's some good discussion to be had here that perhaps it's not as simple as sort of waving those things away if we drill down into the underlying studies, thinking about things like consistency, strength of association, temporality, and so forth. That group of researchers very usefully proposed what they refer to as a new paradigm, a different paradigm, which is they and a number of other research groups, including my own and many others, have gotten really interested in, and that's the notion that, in fact, inflammation may precede disease. So classically, it's a consequence, but in fact, inflammation may come further up the causal pathway and be if not a cause, although perhaps a cause, at least a contributor or an antecedent or precede disease or less milk, which is it's a really useful hypothesis, and lots of groups are gathering evidence around that. What I would point out for today is that there is both evidence and gaps in knowledge on both, relative to both of those sets of hypotheses as to what may come first, what may be chicken, what may be egg, what may be cause, etc. So for example, if the notion is that diseases like mastitis and metritis lead to inflammation, which then leads to lower feed intake, higher NEFAs, higher BHB, etc. Certainly there's evidence that would be the case some of the time, but there's also evidence that in some cows, for example, decreases in feed intake precede disease, and so on and so forth. So again, like the blind men around the elephant, I think we've got a number of people working away on this, and we don't have the whole picture yet. I want to address a few of the challenges that were laid down in this review and another more recent one. And that's, again, trying to understand both the pros and cons, the strengths and weaknesses of these different threads of evidence as we try to answer different questions or different types of questions. And so one of the critiques or discussion points was that a number of these observational studies, many of which have been prospective cohort studies, were portrayed as perhaps ascribing cause and effect when in fact that's not really the case. And so I went back through and really didn't find much indication in any of these cited papers or a number of the others that sort of said, ketosis causes this or that, for example. Although it's probably worth pointing out that there may be some confusion or contention around the use of the word effect. So in a strict or narrow sense, causes have effects, and that's something that could be established in an intervention study at a controlled experiment or randomized controlled trial. And there could be a distinction made between an observational study, say a prospective cohort study where there's an exposure, say ketosis, that is associated with a subsequent outcome, whether that's the incidence of disease or the level of milk production and so on. There might be a little bit of confusion because the measures of association from an observational study or the measures of effect from an intervention study are in fact the same metrics, odds ratios, risk ratios, even kilos of milk and so forth. And so the unit of measure or the measure of the association or effect can be in the same currency or the same units, which may lead to perhaps to some confusion. Another point of contention has been that there's inconsistency across studies in various cut points and strengths of association and so on. So again, one of Hill's criteria was consistency. So that's a fair point. But generally, philosophically, since apparently that's my gig, you expect variability, some variability among studies, because each study, whether it's observational or interventional, is a random sample of the big T truth. More to the point, more practically, different outcomes, you would expect to have different cut points, different measures of association. So the association of hyperketonemia with the risk of DA or with culling, you wouldn't expect to be the same as with the amount of milk at peak or through the whole lactation. They're different biological outcomes. And again, there's this, back to the blind men around the elephant. It's not the case that controlled experiments or experimental models that could reproduce a metabolic condition like hyperketonemia is at the pinnacle of the evidence pyramid. In fact, there really aren't any experimental models for most of the important health disorders in transition cows. We don't really have experimental models for retained placenta, for lameness, for metritis. We somewhat do for mastitis. But even if we did have really precise experimental models to reproduce these conditions these disorders under controlled conditions, it would be practically impossible to do that sort of thing at the scale, the sample size that's necessary to look at dichotomous outcomes like disease, yes or no, premature culling, yes or no, pregnant, yes or no, like Daryl talked about earlier. And so again, there was some critique about these which measures are used, but as Daryl talked about, the measures of association or effect are inherent to the study design, to the type of data that's collected and how it's collected. And so again, that's these measures, odds ratios, risk ratios, relative risks, hazard ratios, are inherent to the data and are correctly applied accordingly. However, one of the constructive criticisms is that at least some of the time, observational studies or intervention studies could do a better job of presenting not only relative measures of association or effect, like odds ratios or risk ratios, but also, not instead, but also having absolute measures of effect. So not simply saying the odds ratio of ketotic cows having displaced abomasum equals 2, but saying that means 6% in the ketotic group and 3% in the non-ketotic group, because for practical application, that may have a different meaning or a different implication or application if that odds ratio of 2 is a risk of 1% versus 0.5% as opposed to 10% versus 5%. And again, there's been a lot of talk about what's a dogma, and I think we again are all in strenuous agreement that dogma of any sort is not helpful to advance science or management of dairy cows. Again, there's this much more recent review paper from Rico and Barrientos Blanco, just published a few months ago in Journal of Dairy Science. And again, really constructive, very comprehensive review of biology and a great many studies, very useful contribution to knowledge and to the debate. Some of these points I've already talked about, but they also build the case for inflammatory disorders, perhaps associated with fat mobilization, actually being upstream in the causal pathway. And I think that's a really useful hypothesis for which there's some good evidence. But again, if we come back to which blind person around the elephant are we, what is causal is not necessarily useful, and what's useful is not necessarily causal. So a marker, be it hyperketonemia or something else, can still be useful even if it's not tightly tied to an upstream physiologic pathway. And again, there's a fair criticism that treatments for ketosis have been a little bit disappointing, particularly in terms of improving milk production. That's true, but it doesn't refute the fact that there's quite a lot of consistency and strong associations with reducing the risk of disease and premature culling. And again, I'll come back to that in a second, but if we get tunnel visioned on milk, we're maybe missing part of the picture. And last but not least, there's a lot of discussion about a whole body of work whereby ketones have been suggested to have anti-inflammatory properties, where it's well established that ketones are an alternative fuel that spares glucose for cows to make milk. So making ketones is physiologic and necessary for cows to make lots of milk. a number of the studies that suggest they may be anti-inflammatory or have other effects that would seem to run in different directions to the classical, or if you want, dogmatic evidence that we've seen in dairy cows. Well, a lot of those are not from lactating dairy cows. They're not from transition dairy cows. They're not from dairy cows at all. They're from humans and rodents, none of which were lactating. And so it's tough to extrapolate that again back to external validity. And so as we wrap things up here, again, there's a really nice review paper published by Jess McCart and Sabina Mann last year in Vet Clinics in North America. I think the title, is ketosis a disease, a marker of a high-producing cow, or both? Again, it depends. But there's conditionality there. So, and some of those it depends is in terms of the timing of the measurement or the onset of hyperketonemia, the level of blood glucose of the affected cows, how long the hyperketonemia lasts, etc. There's conditions to those things. So it depends, but that doesn't make it impossible to make sense of things. And so it's, our ability to understand these things is maybe not just a big mess. It's complicated, but it's not impossible. And it's a work in progress. And again, experimental challenge studies can be really useful, but we really don't have a good model to induce hyperketonemia consistently in transition dairy cows. Some of the classic work that was done in Jim Drackley's lab more than 30 years ago did recapitulate reduced feed intake and milk yield, but paradoxically, even though they were feed restricting cows and running ketone precursors into their feeding them in this case, as opposed to infusing them, but paradoxically, they didn't actually reproduce hyperketonemia, at least in that study. And there's a really nice comprehensive review of a lot of these studies with their strengths and weaknesses in that recent review paper. I want to mention briefly a different context here. So we'll take a quick zip over to hypocalcemia as opposed to hyperketonemia. But the point here is there's a really nice experimental induction study that was done by Natalia Martinez and in Jose Santos' group, published 10 years ago. And the question was, does hypocalcemia impair neutrophil function, immune function? And so there was lots of observational study that indicated such an association. They wanted to take it to a more mechanistic controlled experiment. And so they took 10 cows and ran an intravenous calcium binder into the cows over 24 hours and really nicely recreated subclinical hypocalcemia. And the headline is that indeed that did cause impairments of neutrophil function, phagocytosis and oxidative burst. However, they did it in non-pregnant dry cows whose mean parity was 2.5, as opposed to actual transition cows, and in particular, older multiparous cows, which arguably would be the population of interest. But that makes things a lot more challenging because those animals are inherently more variable. The other thing to point out, this is from the same study, when they induced subclinical hypocalcemia for 24 hours, the cow's feed intake dropped for the day afterwards. Their blood calcium obviously dropped. Their blood glucose also decreased. Their plasma insulin levels also decreased. Their plasma NEFA levels increased. So even in this really elegant, very well-controlled experimental induction mechanistic type of study, we're sort of left to wonder, well, is it the calcium that causes the impairment of immune function? Is it the reduction in feed intake? Is it the reduction in glucose? Is it the increase in NEFA? Is it all of the above? Is there a complex relationship of all of the above? And probably so. So one of the other critiques, if ketosis were a problem, treating it should produce more milk. Well, that would sure be desirable. But the fact that our treatments are less than perfect doesn't negate the fact that the condition or the disease, if you want, is a problem. See mastitis. Mastitis is, clearly not a desirable thing for cows, with lots of associated undesirable outcomes from less milk to culling. Our treatments of mastitis are far from mitigating all those consequences, but I don't think anyone would argue that mastitis maybe isn't worth worrying about for that reason. And similarly, whatever disorder we're looking at, but say back to hyperketonemia, milk is important, but we've got survivor bias in there, among other things. And so to finish up here, we started out with the Socratic question, is it useful? Which may also be the manager's question, is it useful? We still want to know what causes what, but along the way, or in the meantime, the questions may be more, is it useful to track the prevalence of hyperketonemia in my herd or my client's herd, because that may inform me as an early warning system, or give me a piece of the puzzle as to what's going on in the herd that I wouldn't otherwise have. It may be beneficial to test and treat individual animals with the goal of reducing their risk, or the herd's overall incidence of bad things like DAs and premature culling that are associated with hyperketonemia. And we're getting slowly smarter and more refined and more informed about which herds and which cows under which circumstances may benefit from implementing these programs. So to finish, mechanisms and causality are interesting. They're important. They help advance science. They're not necessarily required for us to identify useful actions for the field. Experimental models are really desirable to have, but in fact, we don't really have them for hyperketonemia, or in fact, for most of the transition cow disorders that we may struggle with. Well-designed, sufficiently large, well-planned, well-executed randomized controlled trials are near the top of our evidence pyramid to inform what we do as managers, advisors, and for implementation in the field. Observational studies also have their place, and in fact are necessary for us to understand the consequences and associated effects or downstream happenings of cows with health disorders, and properly understood, they also help advance science. And so with that, on behalf of both Daryl and I, thank you for your attention. It's been a pleasure to have this discussion today, and we look forward to taking some questions from you.
All right. Thank you very much, Drs. Nydam and Dr. LeBlanc. That was very interesting. I'm kind of curious, when is the midterm scheduled for? That was quite deep, and I think we'll have quite a few people that'll want to tune in to watch this over a couple times. Myself specifically, I don't think I captured it all, but anyway, before we get started answering the questions, we'd like to share a brief video, and then we'll be right back to answer the questions submitted during today's presentation.
Delivering the perfect ration for the rumen microbes might be more important and more challenging than feeding the cow. NitroSure precision-release nitrogen delivers a consistent supply of rumen-protected nitrogen to improve animal performance, maximize profitability, and minimize nitrogen excretion into the environment. With NitroSure, you get improved fiber digestion, increased microbial protein production, and reduced dependence on expensive protein sources with a high carbon footprint. Feed the microbes that feed your cows with NitroSure precision-release nitrogen from Balchem. Visit balchem.com to learn more. As a reminder, you can still submit questions to the Q&A tab at the top of your screen.
So, gentlemen, our first question comes in from Roberto. Do you feel that p-value cutoffs, like 0.05 for significant differences and zero 0.1 for trends, are appropriate, or can they be too stringent in certain circumstances?
Do you want me to jump in, Stephen?
Go for it.
The short answer is that any bright line cut point is probably not always correct. There's too stringent in certain circumstances, and in others, not stringent enough, right? I think we should use p-values for exactly what they are: the probability that the observed differences are due to chance given a specified model, given the null hypothesis is true, blah, 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 blah. And it tells us specifically what it is. And then it's all about gambling, really. Do you want to accept that risk with what you do next? If it costs you a lot and the reward is going to be very small, I'm going to want a really small p-value. If it doesn't cost me much and the reward is big, I'll accept a higher risk for that. Just think of gambling. It's a probability.
Yep.
Yeah.
And I would just add to that. I would agree with Daryl. Yeah, useful number to look at, but never in isolation. It's not the judge. It's not it's not clever to say, aha, p was 0.06 or 07. Therefore this difference is not real, or something along those lines. It needs to be looked at in the context, among other things, of the sample size. If a study was too small and then we say, p is some big number, therefore there was no difference, but that may well be a type 2 error. In other words, you should have found a difference but didn't, because you didn't have the power, or at least that's the risk. And so it's interesting to point out too that the debate and even trend in science right now towards not just drawing lines at p less than x is a difference and p more than x is not a difference, is actually being driven not by animal scientists or veterinarians who are trying to color outside the lines, but in fact by statisticians who are saying, folks, you're getting this wrong more, way too often. We need to not just draw bright lines and be, dare I say, dogmatic about them.
Mm-hmm. Very well. Dr. Nydam, you mentioned gambling, and I found myself thinking during your presentations, boy, I wish there was more of a practical indicator, right, that kind of takes into account type 1, type 2 errors, costs, all those things. And what about something like gambling odds? Is there any thought to maybe being able to develop something like that to give the practical nutritionist, dairy farmer some guidance?
The most practical guidance I'll give to the veterinary practitioner, the practicing nutritionist, is to get a hold of Stephen's phone number, and if you have that question, give him a call. He's a nice guy. He'll help you out. So odds, you mentioned odds or risk. Those are measures which we often use, and they often should be interpreted along with the confidence interval around those numbers, and probably the p-value too. And as Stephen very nicely pointed out, the absolute magnitude of those differences as well. So yeah, we got to keep it practical. And the most practical thing to do is to look at all these measures in combination in regards to the answer that we're driving at.
All right, while we're talking about p-values, how big does a study need to be to have when a meaningful number when assessing milk reproduction or disease outcomes?
It depends.
And
so no, but there's a little bit of arithmetic, and this is not black box stuff, but the size of a study, generally speaking, and Daryl talked about this when he talked about, how wide is a confidence interval around some number, some measure. It's very much related to that, is how big of a difference are you expecting to find, or to put it another way, how big a difference would be important to find. I mean, do I care about 1 pound of milk? I mean, I might. I'd care a lot more about 5 pounds of milk. But so how big a difference is reasonable to expect or would be meaningful or economically important? And that could be in pounds of milk. That could be in percentage points of pregnancy rate and so on. How variable is the response or the difference between the two groups going to be? And those are the two things that drive the sample size. So again, there's no sort of one number, but if you want something really for the back of the envelope, if we're talking about disease risk, culling risk, pregnancy risks or rates, it's safe to say that you minimally need hundreds of animals per group, maybe many hundreds of animals per group. Generally speaking, for real life cow studies, if our outcome is pounds of milk, we would need fewer cows than we would for pregnancy or culling or disease. But as we get into looking at fresh cows, where the variability, the standard deviation, is going to be bigger than it might be in mid-lactation cows, well, then we would need a bigger study there than we might for something else. So again, there's no one answer, but these things aren't magical or black box. There are many free online calculators that you can plug some numbers into and get an estimate of how many cows would it take if I want to look at this or that.
Oh, okay, good. Next question comes in from, and I may get this wrong, and Noah. And the question is, any comment on using exposure variables as continuous or categorical, in parentheses, BHB concentrations versus hyperketonemia in a prospective cohort study?
I can take a first stab at that, and then Daryl may want to add. So, okay, there's a principle that says that whenever you take data that are inherently continuous, say a measure of blood BHB, it goes from 0 to 6 or 8 or 10 millimoles per liter, and using the handheld meter, that's in 0.1 increments. That's sort of the scale of measurement. So it's continuous. There's a principle that says whenever you take continuous predictor data and distill it down into categories, 2 or more, you lose, at least potentially lose, some information. So the first answer would be, it would often be desirable to leave continuous predictor data as continuous, because potentially you can gain more insight and have sort of more, better, different analyses by looking at it that way. However, when we talk about doing cohort studies, for example, you have to put animals into groups. And so it's useful and valid to use classifications, cut points, groupings that have been well established in other contexts. They're not just made up. You don't pull them out of thin air. You don't necessarily just sort of cut the data in the middle or in the top or some other place. I mean, those are all things you might look at by way of exploration. But the point is that if and when we put animals into groups, they mustn't be arbitrary or made up or kind of putting your thumb on the scales, but rather because they're actually based on the present data or previous data that say this is valid, this is associated with an outcome that's relevant here. So again, it's based on data. We're not kind of just making stuff up or cherry picking.
Very well. All right, I see we're about 10 minutes past the top of the hour. I would like to close with just one final question. And this one comes in from Carrie. What is the biggest mistake you see in dairy-related experiments? Low sample numbers, poor trial design, or overinterpretation of results?
All of the above.
All right.
All of those are potential things that you should look for, and they don't happen in all studies. but to some measure, some of those things happen in a lot of studies.
All right, very well.
Including ones that I've done, frankly.
All right. Well, I want to thank both of you. I also want to thank everyone for attending today's webinar. If you have any additional questions, please submit them to anh.marketing@balchem.com. The Real Science Lecture Series continues with educational topics each month, including our next one on September 10th with Dr. Frank Mitlerner, and he'll present Sustainable Livestock, Sustainable Future. Why we need a toolkit for solutions to improve sustainability in animal agriculture. Our schedule for the remainder of 2024 is now available online. You just visit balchem.com/realscience for details on those webinars and to register for upcoming events. Balchem's podcast series continues to offer a deeper dive into our webinar topics. Log on to your favorite podcast platform and search for Real Science Exchange, or visit balchem.com/podcast. If you want a really cool Real Science Exchange t-shirt, just submit it to Real Science Exchange and send us a screenshot along with your address to anh.marketing@balchem.com. We'll get that to you right away. On behalf of Balchem, Dr. Nydam, and Dr. LeBlanc, thank you for joining us today.